since it didn't. So once you have read all the papers, what do you get? You get something like this. Which is like, this represents about 10% of all the pathways that we know of are implicated in cancer. Yeah. So that's pretty complex. And this is generated by reading papers in an automated sense. So the question is, once we have something like this, what do we do with it? Of course, if we are very sure, using all the things that James was saying, pinning down the reliabilities, removing conflicts, uncertainties, and everything.
Is it still moving?
It's still moving, but don't worry about it.
I'm not just
So what do we do with it? So that's the question. How do we use this kind of thing to design interventions, or essentially maybe go towards a cure? So that's what I'm going to talk about once we can make it stop moving. Okay, so to go back at the front, the issue which James spoke quite a bit about, pretty much the central issue, is uncertainties and conflicts. So again, it's probably just a repetition, but conflicts arise from many different sources. It's hard for computers or machines to read papers written in human languages. There's also experimental errors. And sometimes we do not know, nobody knows. The researchers do not know what the underlying molecular interactions are. So essentially, the best we can do is guess. So that is also something that's there. And one thing that's also important to note, if you have seen this, what I just showed you, this huge network, biochemical interaction network, all the metabolic pathways that biologists are definitely familiar with, those are pieced together from many, many different experiments. These experiments are subject to different experimental errors, different conditions. There's kind of a little bit of wishful thinking to say that all this pieced together, we understand this huge complex system. So that's also an issue. And the way to know whether we are getting any closer to understanding this huge complex system is to generate hypotheses from what we have read and validate that in labs. So that's what we're going to go towards. But it's not always easy to combine experimental data. And when I say experimental data in this entire talk, I mean essentially expression profiles, so expression data, with what we have read and make the predictions better. So it's not always easy to combine these two kinds of knowledge. And the reason is this. When you're talking about, particularly when you're talking about expression data, you don't the data itself doesn't tell you whether, say, gene A activates gene B or gene B activates gene A, or it doesn't actually say that. It just tells you what you see in expression. So if you have you have read something like this, that A inhibits B or A activates B, and you see when you see a simultaneous expression, you see something like that, you can't really say from that B is inhibiting A. You can't, because they're not operating in vacuum. They're part of this giant network. So it may be that A is suppressed because of the effect of other entities in the network. Similarly, in the other one, you can't really say that A is suppressing B. So it's kind of non-trivial to see how you can combine them to remove conflicts. Of course, you can get something. For example, if you have this, where both A and B are expressed, it's unlikely that A is inhibiting B or B is inhibiting A. So there are issues with it. It's not obvious, at least to me, how, like, I'm just making the problem. I'm going to talk about how to go towards solving that. But it may be not obvious how you're going to combine this kind of two different kinds of knowledge and essentially make better predictions or go towards a more complete understanding. So it may be easier to think, instead of thinking about biochemical networks directly, it's somewhat easier to think in terms of influence networks, which are just nodes and which pass on influence to their downstream nodes, which pass on influence to other nodes, and so on. So if you think the nodes as biochemical entities, genes, and which can produce gene expressions, produce proteins, and so on. So if there's an activation of one node, as I said, it activates downstream nodes, it activates other nodes. We're not discounting feedbacks, that can happen too. So in that kind of scenario, if you're given an expression profile, say chi, I don't know whether you can see it, which is a vector, so the ith entry, say, represents the magnitude of the expression of the ith node, then how do you go about modeling this flow of influence in this network? Well, if you assume that this influences occur with certain probability, so there is a probability transition matrix which represents a network, one-step influence, say that transition matrix is pi, then one-step influence would be just pi into x. Two-step influence would be pi x plus pi square x, and so on. So essentially, you can represent these flows by the sum, where on the left-hand side, the nu is essentially this combined flow, and chi is the observed expression data. So if, and note that when you are observing an expression profile in a system, that is in some sense an equilibrium state, because you probably won't be able to see this gradual change of influences. Well, you can probably design a complicated experiment to do that, but in standard expression profiles, you don't see that. What you see is like a equilibrium snapshot of a system, which means that if the structure of your network is correct, and if there's no errors in the expression in the experiment where you got the expression data from, then the chi and the nu should match. And that, why am I talking about this? This allows you to essentially define how you can, as I said, the problem was how you can combine those two kinds of knowledge, a network, that initial network that you have read from papers and the experimental data that you have. How do you make them, combine them together to get something more? So that kind of allows you to define the optimization problem. Essentially, it's simple. We want to tweak the network so that the left-hand side, the left-hand side of that matches the chi, right? So that's essentially the optimization problem. And note, what you can change, of course, you can't change the chi, that's expression data you got. What you can change is the connectivities in the network. You can tweak them based on all the values identified by doing all the work that James talked about. So there are probabilities, reliabilities on the links. But given that, if we can tweak it so that the nu and the chi approach each other, then we can say that the expression data that we observe is being more closely represented with our inferred interaction network. Now, that seems easy. The issue is that biochemical networks, as I said, are much more complex than that. For example, they're not just influence. Nodes or genes, at least, you can't just represent them just passing on influence to the downstream nodes. In the least, you have to talk about whether the effect is inhibitory or whether it activates something. And in many cases, one gene can activate something and inhibit something else. So the situation is much more complex. And so to explain it, let's talk about a very simple situation where, say, a gene Q1 affects another gene Q0. So suppose it activates it. Now think about that for a moment. So how do you write down the probability of expression of Q0? That is essentially the probability of the interaction occurring multiplied by the probability of expression of Q1. So if you think about it, the way to represent that as an influence graph would be to write the edge in the other direction, right? Because then what is the probability of expression of Q0? That would be just the probability of the interaction multiplied by that, so the arc goes the other way. Also, as I said, in the least, you have to talk about the positive and the negative effects of each node, which means that each biochemical entity has actually two images in this influence network, the positive effects and negative effects. So it looks kind of complicated, but to think about it, the simplest way of representing such a biochemical statement is by something like this, where each node has two images here, and the interaction is specified by a virtual node. So So if Q1 is positively interacting with Q0 or activating Q0, then the picture looks like something like this, where essentially you're switching off the connection to the negative impacting part of Q1. So why does that represent a positive regulation? Well, look at it, look how this will work out. If Q1 expression goes up, then what happens to the expression of Q0? Well, the probability of this and the probability of that into the probability of the expression, so this one goes up as well. Similarly, to represent negative regulation, you have to turn off the other arc. It gets more complex when instead of having this kind of simple interactions or direct one-to-one interactions, you have one entity regulating a reaction. And again, it follows kind of the same principle. Each entity gets represented by positive and negative images in the influence graph. But to capture the notion that when expression of A goes up, the reaction goes through. If there's a positive regulation, goes down, it stops or proceeds much slowly. Essentially, you have to model a switch between more than one virtual node. I'll not go into too much details of that. So why am I jumping through all these hoops? As I said, influence networks are much simpler, mainly because they're just digraphs, right? There's nothing really you have to write on the arcs to explain what the network does. It's just nodes and edges. Whereas biochemical networks are not like that. There are different kinds of interactions, positive, negative, many other kinds. So there's a lot of annotations that you have to write down on the edges to fully explain the information that is representing. And that's a problem because all network science that people do, particularly, say, in communication networks, there's a lot of literature on that, a lot of optimization algorithms there. Most of it deals with digraphs. So if you have a network where you have to write stuff as annotations on the edges, that's a little bit problematic. And if you could take that and somehow represent it as just a vanilla probabilistic digraph, it opens up a lot of possibilities for optimization. So that's essentially the reason why I'm jumping through all the hoops. And I'm not going to talk about, and the optimization problem that we're solving is essentially moving that flow as close as possible to the expression profile. And there are multiple ways of doing that. That is one of them. And so let's talk about what kind of data that we have. So essentially, all this is about grounding data, right? You read the initial network from the papers, and you have expression data from experiments, and we are essentially combining them. So those are the kind of sources that we have. I'm not going to go too much into them. One of the questions that comes up is, by doing all this, what happens if the expression profile that you get is not from a real system? That's kind of a nice validation experiment one can do. What if I give you random expression data? How would this whole system work? Well, it shows up clearly that you don't have real data. So if you have real data, the deviation between the flow vector and the expression vector, which can be represented in degrees, and smaller the better, is something like this. Whereas if you use random data, it's clearly different. And moreover, if you optimize this pre-optimization, running this optimization algorithm, and if once you do the optimization, you push it even more to the left. So that kind of clearly shows that, well, if you have actual expression data, you have a very different kind of distribution with much smaller error. If you have random things, you don't. And also, you really cannot optimize the system at all, pretty much, if you have fake data. So this is what you end up having. And we have many different expression profiles, which are coming from many different sources, from many different subjects. And if you read an initial network like this and ground it against any or all of those expression profiles, you get a kind of optimized network, right? Which already eliminates a lot of the conflicts. However, the conflict may still remain. So if you zoom in into that portion that was encircled, you'd see that the network is not exactly matching up. Here, the green is a positive regulation, the arcs, and the red is a negative regulation. And you see this, there are differences there. So there are conflicts. However, now it's much simpler to resolve them. Since we have a notion of how well particular expression profile supports the initial network or the optimized network, we can remove the conflicts by focusing on those situations, only like increasing the weight on those situations where that deviation is low. So essentially, you're saying that if you give me an expression profile where I am not being able to reduce the deviation between this the flow vector and the expression vector by too much or at all, then the expression data probably does not represent or did not come from such a network. So something was wrong with that. So if you do not account for those, if you account for those and do not use them to make your make the judgment of how well it's matching, then your kind of conflict goes away. So that is a nice way of doing that. So again, going back, what do we gain? Well, what we're trying to understand here is this big mechanism underlying the processes. And more importantly, if you have something like this, which we are kind of confident is what's going on because we read from the papers, we grounded against experimental data, then that gives us actually a huge model of this complex system. And since we have this network, we can now go towards understanding perturbations, right? And that is important. Perturbations are on the whole network. So if say this is activating, this gene is activating that gene, then it's kind of obvious what happens if you alter this. That gets altered. That's obvious. But what happens when you alter this? What happens somewhere here? To know that, you need models like this. And that is kind of very useful, because that allows us to essentially design what will be the effect of drugs, which are small molecules affecting certain targets, blocking targets or activating targets. Again, for individual drugs, the peripocine, for example, it has, most of the time, it has a known target, ATK here. So you know that if you introduce peripocine, ATK expression is blocked. That's kind of obvious. What happens to the rest of the network? That's an interesting question. That is kind of non-trivial to answer just from the experiments which come up with the fact that peripocine blocks ATK. And more importantly, what happens when you mix two drugs? Again, the two drugs has their own targets. We know what's going to happen there. The question is what happens elsewhere. And we can test that by doing this huge model. We can take out the connections. We can simulate the effect of different kinds of doses, and we see what happens. And it's kind of non-trivial. And you can also see what happens if you increase the dose. See, the effects do not add linearly. You have this at a low dose, increase the dose, becomes this, then this. At this point, the cell is dead. So let's talk about a very specific example of a triple-negative breast cancer. This kind of breast cancers where the cells do not show any estrogen or progesterone receptors, and there's no HER2 expression, right? And in this case, the tumors grow aggressively. And the problem is, since there are none of those hormone receptors and HER2, hormone therapy is not helpful. So the only way of handling these things is chemotherapy, which is not a great option. So essentially, it was Andre's idea. Since all these receptors are missing, that's not normal. That is kind of abnormal behavior of a cell. So what if we try to force expression of, say, an estrogen receptor? Will that move the cell towards a more normal operation? Will that slow the disease? We don't know that. That's still we have to do experiments on. But let's try to find out first if by using this model that we have, essentially we have all these pulleys, by tugging and pulling in the network, we can actually force expression of a particular gene, say a estrogen receptor. And we can only look at this pulleys, this levers. What I'm talking about is levers. I mean those druggable targets. So if you look at this all different druggable targets and see whether that increases, say, ESR1 expression, again, that is not that difficult. If you have some gene inhibiting ESR1 expression, you take that out, of course, that will increase. The non-trivial part of the problem is the y-axis, where you have to minimize the effect on the rest of the network. If you could do that, if you could just increase the SR1 without affecting anything else, yeah, that would be great. So can we do that? So we go into this and we look at different kinds of targets which can be blocked by known drugs or known small molecules, and see what happens. What is the effect on this in this kind of trade-off plane? And we get some results. These are when you use the drugs by themselves, right? But as I said, by having this model gives us the ability to go and look into what happens when you combine things in different doses. So we have not done this, it's kind of work in progress, but we have looked at what happens when you combine pairs of things. And you see what happens. This is the ESR expression. Some of them are bad, doesn't go up. Others are pretty good. Again, that's not the only thing. You'd also look at what happens to the rest of the network. So more yellow here is better. And that is the perturbation in the rest of the network. So more blue is better. That kind of goes into telling you what things is expected to work, right? You have to choose something very yellow from here, and then to make sure that it's very blue another thing. Now, it's kind of, the whole computational part is rather complex, right? You're only talking about a two-drug cocktail here. And that's already kind of quite intensive to do the optimization, figure out this kind of Pareto front and all that. Now think of if you want to go 3, 4, you're actually talking about millions and millions of possible combinations. So it's kind of intensive. So that's what where we are trying to go to. And I'll just conclude here. We're trying to unravel big mechanisms. We're trying to optimize this kind of therapeutic cocktails and trying out some new ideas which may be useful for intervention in complex diseases. And what are the implications of machine science? You want to talk about it?
Oh, me?
Yeah.
Yeah. I mean,
You're the one who has the paper in science about machine science.
These have implications for machine science.
So that's acknowledgements, and that's Andre. Okay. So.